Biotech Therapeutics. We are a biotech company located in the north of Spain, and our vision as a company is to be able to provide novel effective treatments to help cancer patients, which we all understand is a big medical need nowadays. So, well, the problem when we're thinking about developing compounds and novel treatments to cancer is that, as you can see in the slide, most of the compounds that are developed actually don't make it through the clinical trials and approval. And actually, the chances for approval of a compound are less than 5% in oncology. So basically, most candidate drugs fail to reach the patient. And this is causing two major problems. First is that research in pharmaceutical industry has an outrageous cost, and then obviously we are not able to fulfill this promise of giving patients novel compounds. And if you go back to how compounds are actually selected and how to make decisions about which compounds to pursue in clinical and pharmaceutical development, we can see that almost all, if not all the drug screening campaigns are identifying compounds based on experiment carried out on conventional 2D monolayer cell culture. So the way we decide whether a compound is interested or not is based on a very obsolete cell culturing technology, and this is conditioned in all the process because once the hit is identified, then they stick to that hit until it fails. But the problem is, as we know, that tissues, organs, even tumors, they are not 2D structures. They are structures that grow on 3D, and biology is not flat, which is actually the title of the talk. So we need systems that recreate this 3D architecture in vitro, so we are able to better understand how a compound will react to, how cancer cells will react to compounds to make better decisions on which compounds to actually pursue for pharmaceutical development. So the answer is we need 3D cell culture systems to recreate this. So this is, as Peter said, that nowadays I think mostly everybody is familiar with the existence of 3D cell culture. And but I'm not sure, I'm not convinced that everybody understands what is 3D cell culture or where are the barriers to actually implement 3D cell culture technology in their developments. This is in the slide, this is a slide just showing some data comparing the effect of several compounds in a triple-negative breast cancer model. As you can see on the left, an example is geldanamycin, which is a compound that interacts with HSP90. And as you can see, the profile of the compound in 2D was very beautiful. Actually, you will see this profile and you will immediately choose this compound as a potential candidate. However, if you did the same experiments using 3D systems, you will see that it didn't have any effect. Actually, if you follow the clinical development of geldanamycin, it failed in clinical trials. On the other hand, you can see other compounds like palbociclib, which is a CDK4/6 inhibitor, and it didn't have a very attractive profile on 2D. Now, if we do the same experiments in 3D, we will see that it has a beautiful activity, and actually palbociclib was likely approved for, pushed for fast approval by the FDA, and a few months ago it was approved as a medicine. So this is two extreme examples on how 2D and 3D cell culture influences the way we make decisions in pharmaceutical development. So as a summary of the advantages of 3D cell culture is compared to the in vivo situation, and by in vivo I mean the physiological situation. Well, first of all, morphology. A cell in 2D monolayer culture is a flat structure, which is not the structure that we see in vivo, and 3D cell culture actually recreates this cellular morphology. On the other hand, in a 2D monolayer flat culture, the cell is in contact with a non-physiological substrate, being an extracellular matrix, artificial matrix, or plastic. In a 3D cell culture system, this doesn't happen. Cells are in contact with their own other cells and with the matrix that they secrete. These conditions, the way cell contact with the other cells, and this is typically absent in 2D monolayer cultures, obviously depends on the confluency, but these contacts are preserved in 3D cell culture and obviously in vivo culture. 3D organization obviously is not present in 2D monolayer, which is preserved in 3D spheroid and is preserved in vivo. And another point that's very important is how cells interact with the medium, the nutrients, the oxygen, and obviously drugs, because this interaction is abnormally high in 2D monolayer culture and is close to normal in 3D cell culture and obviously in vivo, which is the standard. And there's been many papers in the literature describing how gene and protein expression signatures and biomechanical behavior of cells in 2D is very different from the normal situations. And indeed, 3D cell culture preserves this gene and protein expression signatures and biomechanical behavior, which in this slide what we pretend to say is basically that 3D structure are much better predictors of how the cells will react to particular compounds. I'm talking all the time about 3D spheroid culture as 3D cell culture. There are different forms of recreating this 3D architecture in vitro. However, basically can be divided in two, which is scaffold-free and scaffold-based. But I'm most of the time just looking into ways that we can recreate this structure in a way that is robust and effective for drug screening. I understand there's many very sophisticated systems that may recreate the 3D structure, but they are not amenable to drug screening. So what we're trying to do that are easy enough, allow manipulation so you can do drug screening. You need to perform hundreds of thousands of experiments, and so you can select compounds. And these systems are typically known as spheroids or 3D spheroid systems. And this is what I'll be talking about the rest of the talk. So today is cell-based drug screening comparing 2D and 3D spheroids, and there are several ways of doing spheroids. And the standard right now is the 3D aggregate spheroid system, in which you have cells that you put in a point in space and they aggregate and make a ball, and this is a 3D spheroid. Well, they are not easy to set up right now. I mean, technology does not allow us to do rapid and inexpensive ways of creating aggregates. Biological relevance is of course higher than the 2D. It allows for rapid testing. One, it's optimized and set up, but the throughput is still low to medium throughput. Enriching CSC stands for cancer stem cells, and this is particularly interesting because 3D structures have a higher rate of cells with cancer stem cell or stemness features, and this is very relevant to understand the biology of the 3D spheroids. You can do a viability test on 3D, which obviously you cannot do on 2D, but with a 3D aggregate spheroid system, you cannot do a spheroid formation test, which I'm going to talk next. And talking about the logistics, the 3D aggregate spheroid culture is very similar to 2D assays. It's simple to use, lifespan is short, but it's not easy to store or easy to explain, and convenient and flexible. So the 3D spheroid systems that are based on cell growth instead of aggregation have several advantages. First of all, it recreates the way tumors are initiated in vivo. So you have sort of like a microtumor in a well, and this is a 3D spheroid based on cell growth. And it actually allows to measure two different parameters. On one hand, you can just do a 3D viability test as you would do with a 2D or a 3D aggregate. That's fine. You only have to allow these spheroids to form, then add the compound and measure with the standard viability test. But also, if you are seeing spheroid formation in the well, you can test compounds before the spheroids are formed and then compare how a compound is affecting whether the spheroid is formed or not. So you have another insight into the biology of the tumor, so you can select compounds that prevent tumor formation, and this is very interesting in order to develop novel compounds to treat cancer. So here in the next slide you will see several typical models of different tumor indications like breast, ovarian, glioblastoma, colon or prostate, and you will see in a 20x magnification that the spheroids are very different in morphology from tissue to tissue, and this is what we would expect because different tissues make different structures. And on the 2.5x magnification you will see how you actually will see in the microscope an spheroid culture, so you will either count the number of spheroids or just test the viability on their own spheroids. Now, if you're looking into using 3D cell culture for drug screening, obviously robustness is key. I mean, you need to be able to ensure that your 3D cell culture is comparable from within the experiment and inter-experiments with the experiment you do today and the experiment you do 3 months or 2 years down the road. This is just an example on different 3D spheroid cultures that we have performed in at STEMtech, and just looking at the number of spheroids per well and the number and the size of spheroids, and this is what we're looking for, so a consistency and robustness in 3D spheroid culture. Another important point is what is the biological information that you get out of the 3D cell culture. You can do a viability test, and you can do a spheroid formation test, and you can position compounds here. The dots represent unique compounds on where they stand in these two activities. So you can select for compounds that are killing the cells, compounds that are not killing the cells but are preventing the spheroid formation, and that gives you access to more information and to make better decisions on compound selection. Actually, if we would compare compounds, and these are typical compounds that are widely known on different tumor models, you compare viability on 2D, 3D, and the spheroid formation, you will see that the reaction of the cells is very different in some compounds and is comparable in other compounds. And this is to say that the 3D cell culture is not necessarily different from 2D cell culture for every compound. I mean, it depends pretty much on the mechanism of action, but you will find compounds that behave similarly. But more interesting, you will find compounds that behave very differently, and these are the specific information that you need to select which compounds to pursue. We already talked about yeldanomycin, but you can see differences also in rapamycin or ACD214, that they provide different answers, and these are the answers that you need to select your compounds. Now, this is giving all this information. I would like to introduce to our product, which is by the name CellToSphere. This is a unique and innovative, ready-to-use 3D cell culture solution for compound screening. As a company that have been working in 3D cell culture for many, many years, we understand that the barriers and the level of expertise that you need to implement 3D cell culture is sometimes preventing research lab and pharma labs to actually do 3D cell culture. So we develop a solution that is ready to use, all in one, ready to go. There's basically no training needed. It comes in a kit. Right now we launched the first version of the kit in 96-well format, and it's enriched on spheroid forming cells, which is to say cancer stem cells. We manufacture kits with off the shelf ready to go, but we can also manufacture kits with the layout that you desire to fit your research needs. It's there's no training needed. It's as simple as you receive the kit. The kit is delivered to you frozen. This is, as far as we know, the first solution in the world that is able to provide a frozen solution, so you can just store it in your freezer and use it whenever it's convenient to you. So you don't need to understand, or you don't need to get the expertise in 3D cell culture. Just use a plate whenever you're ready, and if you want to do another experiment, just take another plate out of the freezer. You apply the cell culture medium, which is provided with the kit, put it in the incubator, and then wait 4 to 6 days until the spheroids are developed in the well. And then you have a beautiful 3D spheroid culture in every well to use in your drug screening efforts. So if we can see the continuum of the drug screening process from the early primary screening to the late efficacy validation previous to in vivo selection. Right now we're covering just the secondary biology 96-well based validation studies with our product CellToSphere 96-well. We're also going to be releasing the same kind of culture in 384-well format before the end of the year. And we're also working in another novel system which is going to give a lot of value to the drug validation studies, which is using spheroids not based on cell lines but based on PDX material. These PDXs are patient-derived xenograft grown in mice, which allows to amplify the size of tumor samples that we get from patients. And this has been already reported to resemble pretty much the same gene expression patterns and behavior of the tumor in the patient. So the idea here is with the tumors that we grow in mice, then we get this material and produce plates that are frozen down, and they are delivered to you frozen. So you can select which models with PDX models will suit very much your drug development interest, either by selecting which tumor model will work with your drug, or to select which indication is the best for you, or more importantly, to see the effect of your drug in a close to physiological in vitro kit before proceeding into a long and expensive in vivo system. That's why we call this clinical trial on a kit. This is an example in a colon cancer model. You can see the spheroid number in different plates and the diameter of the spheroids, and these are some pictures showing the spheroids from these colon cancer samples. This is being developed right now. We will be initially providing breast and colon cancer models. And this is just an example of drug profiling using this colon cancer model and the response to different common compounds. And what's more interesting is that when we perform these same experiments, not just in vitro, but with the same model in vivo in mice, we saw a correlation which is around 70%, which for an in vitro system is actually pretty high. You have to think that if you culture these cells into these systems, the correlation will not be any higher than 30%. So as a summary, and compare our cell-to-sphere technology with the 2D or 3D aggregate systems, the advantages is that the biological relevance is higher. The throughput can be adapted from medium to high throughput with the 384 system. And more importantly, the logistical size, as it is a frozen product, it's very simple to use. It's a longer lifespan because you can use it for 6 months, and it's easy to store and easy test planning, convenient and flexible. And these are the main advantages that we provide with the cell-to-sphere 3D spheroid system. And now, like I say, we're providing several tumor models in several indications. Those are conventional cell lines, but they have been validated in our system for robustness and viability after freezing. We can also manufacture plates to suit your needs in terms of these cell lines, but we can also try your cell lines and make plates with your own cell lines, whether you have a particular cell lines that we haven't worked with, or you have a cell line that expresses or expresses some particular gene or reporter, we can put that into our system as well. And then later in the year, we'll have the 384 well and the PDX systems coming to the market. This is our contact information. You can engage with us in social media, and then you can contact Bilo Biotech for also for sales information. Well, thank you very much, and I'll be very happy to take your questions.
Thanks a million, Angel. I think it was a great presentation, and a few questions came in. I will start from the top. There was a question, I think, whether you can also use, or have ever tried glioblastoma primary cells so far.
Yes, we are working with some glioblastoma samples in the PDX system, and actually glioblastoma is one of the easiest models for spheroid formation. It was one of the first described. Yeah, basically because it comes from the neurospheres models, and we have glioblastoma cell lines available for ordering right now. Yeah, those glioblastoma is usually a very nice model for 3D spheroid. So I would say that other tumor indications are more complicated, more cumbersome, but glioblastoma is one of the easiest.
Okay, perfect. Another one is, did you ever try a longer culture period than, I think, 6 to 7 days? And why 6 to 7 days?
So the intention with developing cell-to-sphere is that it provides a very convenient, disposable, ready-to-use system. Of course, these are live spheroids, and you can culture them and passage them in as much as you can. However, I would not use an isothermal plate if that was the intention of my experiment. Because then if you let them grow longer, the spheroids will become bigger, and then eventually will aggregate all in the middle of the well, so you would rather use different format. But yes, the spheroids are alive, and you can just passage them and use culture them for as long as you want.
Okay, another one is, what percentage of FPS do you use, if you can tell this?
Yes, of course I can. So this is a defined medium. It doesn't have any FBS. That's part of the reason why the spheroid system works, is that the cells do not differentiate. There's basically the tumor-initiating cells that make the spheroids and their progeny. But it's very relevant not to have serum, because it immediately differentiates the cells and they stick to the bottom of the plate.
Yes, that's right.
So the answer is no serum.
Yes. Another easy question is, coming back to the PDX stuff, so how much time do you need from patient tissue to be able to send the cell to CS plates to the customer? Do you have already an idea?
Yeah, so we are now validating the system in several tumor indications. We hope to be ready for market after the summer with the first models, but the actual launch for most of the models will be towards the end of the year. It basically, we don't have any lead time for that. I mean, we just develop the models and freeze them down and validate. So as soon as the models are validated, we are ready to order, to ship.
I think the question goes more, if a customer will send us tumor material, how long will it take to get PDX and then send the plates back to the customer?
So if the customer sends us tumor material and it's a primary tumor, we can do spheroids with them. We have processed over 500 different samples from human tumors, but the number of plates that we will be able to manufacture will be very limited. If you want to do, if you want to send us just PDX material for around a gram, we can get around 10 plates. Now, if you want us to do the mouse engraftment and all that, then we will be able to do that, but we will have to do a separate proposition. That will be a customized proposition to the customer, and that this will take, I mean, engraftment in mouse, if successful, will take anything from 6 to 10 months, and then there's around 4 to 6 weeks for processing and making the spheroids.
Okay. Do we have any experience whether there is a difference between a classical PDX, that means tumor material in one mice, or after expansion? Because I know that many PDX material is expanded in mice.
Yes, so we have compared several tumors from PDXs, and we do this with partners that already have a lot of experience with PDX experiments, and we haven't seen any difference comparing the gene expression. But I will have to say that that would be up for testing. I know there are papers in the literature describing how close the PDXs are from the original patient tumor, but the way the PDXs are performed is actually not amplifying the same fragment all the time, but you just chop the fragment, just make kind of what we do with the cell lines. We make master banks and word banks, and those are the ones expanded. So pretty much the models preserve the original gene expression signature.
Okay, there's another question. I think that's more or less goes to, I think, new cell lines, or if a customer sends a cell line, can you describe a little bit the process if a customer likes that their cells have to be used for producing sphere plates?
Yes, well, that's the line that we call your own, in which we will be very rapidly be able to see if a cell line makes spheroids and is suitable for freezing down based on our quality controls, and we can do that in 4 to 6 weeks as well. We can produce cell lines to your order, I mean, to suit the customer needs, but we require a minimum plate order to make sense. Otherwise, it will not be cost effective to produce a few plates. We will need at least 20 plates to be able to validate and do this. But that is certainly something we're actually doing.
Okay. Do we have any experience using macrophages within the system?
So we have, we also, besides producing our spheroid kits, we have internal R&D in the company, and those R&D lines are focused into doing co-culturing studies. We are very advanced in working with fibroblasts and cancer cells. We have also a line working with macrophages, but it's something we have not developed very much yet. I mean, we're still a small company. We cannot do everything yet. We will. Sure. But that's certainly something to look into the future.
Okay. I think I have another question which came in, that after you finished an experiment, people might be able to get access to the spheres for dissociation. I think this might be possible using enzymes to get single cell suspensions which can be analyzed in a FACS system.
Absolutely. We haven't done it ourselves because this is not our goal, but we have had customers that have reported back to us, and they have done all kinds of experiments with the plates. After all, it's a live sphere culture, so we have customers that have processed them for cytometry. They have even retrieved RNA and DNA for genomics analysis. Basically, any kind of cell biology experiment that you can think of, you can do.
Okay. I think that we also can provide protocols if somebody is asking for this.
Of course, we'll be able to help you. I mean, our relation with the customer is not just providing a product, but make sure that they have the best experience and they can achieve their goals, so we'll be happy to help.
Okay, that's perfect. And my last question is, you mentioned that new cell lines or new products will come using different cell lines. Can you give any estimation when new cell lines will be available and which will come next?
So we are working with a lot of cell lines all the time. I mean, there are hundreds of cell lines. Obviously, we cannot have them all, but our internal process for validation is very rigorous. So we make sure that every cell line comes in a very robust way after freezing, and not every cell line is actually suitable for that. And besides, some cell lines will just not make spheroids in there to begin with. That's described in the literature. So right now we're working with models that we are not actually carrying, like melanoma, head and neck, skin cancers, and some sarcomas as well. But there are several cell lines in the pipeline that will be available soon.
Okay, perfect. I think that's all for the moment. I hope, and you can send more questions by email, no problem at all, and we are happy to answer these. Angel, again, thanks a million for your great talk, and all the attendees for giving us a time that we can introduce the system and the services to you. We hope to get more feedback from you and wish you a nice day. Say bye-bye. Angela, I think we will talk in the next days.
Thank you very much to you, Peter, and to all the people that took the time to attend the seminar. And we'll be very happy to engage with novel questions and collaborations and obviously customers. Thank you.
Okay, thanks. Bye.